There's a jar sitting in your kitchen right now that the United States military actively uses to heal wounds in field conditions when antibiotics fail. The New Zealand Defence Force issues it as a standard wound care product. The British National Health Service approves it for treating infected surgical wounds that have not responded to antibiotic therapy. Peer-reviewed studies published in medical journals confirm it eliminates methicillin-resistant Staphylococcus aureus, the antibiotic-resistant bacteria killing over 300,000 people globally every year, from infected wound surfaces faster and more completely than the pharmaceutical compounds your doctor prescribes. Ancient Egyptian physicians packed it into infected wounds 3,000 years before the first antibiotic was synthesized. Greek military surgeons carried it onto battlefields across the Mediterranean. Roman legions treated sword wounds with it from Britain to Mesopotamia. Every civilization that kept bees and treated wounds arrived at the same clinical observation independently across 5,000 years of recorded medical history, that this substance healed wounds that nothing else could touch. Then, the $50 billion wound care industry calculated what a $6 jar of raw honey that never expires, requires no prescription, produces no antibiotic resistance, and outperforms synthetic wound dressings in head-to-head clinical trials would do to their revenue. In 2015, the FDA restricted health claims on honey products sold for wound care purposes unless the manufacturer held full medical device registration. Registration costs millions of dollars and takes years. The jar on your kitchen shelf cannot tell you what military field medics use it for. It cannot tell you what the British NHS prescribes it for. It cannot tell you what 3,000 years of continuous medical application and 2 decades of modern peer-reviewed research confirms about what it does to bacteria in an infected wound. The label says natural sweetener. The rest was buried. This is the story of raw honey, the substance that preserved Egyptian pharaohs, that supplied every army in the ancient world with its primary wound care compound, that modern clinical research has confirmed as one of the most effective antibacterial agents ever identified, and that a $50 billion industry spent decades ensuring you thought of exclusively as something you put in your tea. If this knowledge matters to you, the subscribe button and the bell are the only way to protect it before this video disappears. This is where we open the vaults the industry sealed shut. Stay right here. Because by the time this video ends, I'm going to show you the exact military field application protocol the label is forbidden from describing, the infected wound treatment the British NHS uses when antibiotics have already failed, and the documented regulatory timeline showing which financial interests were served when the FDA restricted health claims on a substance with 5,000 years of continuous medical application. If you leave now, you'll go back to paying $40 for a synthetic wound dressing that clinical trials show performs worse than the jar already sitting in your kitchen. Stay here. Everything is coming. Let me take you back to where this story begins. The year is approximately 2700 BCE. In the Nile Delta workshops of Egyptian physicians serving the court of the Old Kingdom, medical papyri that would survive 4,000 years describe honey as the primary topical treatment for infected wounds, burns, and skin ulceration. The Ebers Papyrus, one of the oldest medical documents in human history, contains over 900 prescriptions and references honey as an active ingredient in the majority of topical wound treatments. These physicians were not applying honey because it tasted pleasant. They were applying it because generations of direct clinical observation had established with complete consistency that wounds treated with honey healed, where wounds treated with other available substances did not. What those Egyptian physicians observed but could not explain in chemical terms is now understood completely by modern microbiology. Raw honey produces hydrogen peroxide through an enzyme called glucose oxidase, deposited by bees during production. The hydrogen peroxide is released slowly and continuously at the wound surface in concentrations high enough to kill bacteria, but low enough not to damage surrounding tissue. The low water content of honey creates an osmotic environment that draws fluid out of bacteria through their cell walls, dehydrating and killing them through physical chemistry rather than pharmacological action. The high sugar concentration prevents bacterial growth by binding the water molecules bacteria require to metabolize and reproduce. And then there is the factor that makes raw honey uniquely dangerous to the antibiotic resistance crisis that is killing hundreds of thousands of people annually. Honey contains methylglyoxal and defensin-1, compounds that attack bacteria through multiple simultaneous mechanisms. A bacterium can evolve resistance to a single mechanism antibiotic by mutating the specific cellular target that antibiotic attacks. A bacterium cannot simultaneously evolve resistance to osmotic dehydration, hydrogen peroxide oxidation, methylglyoxal disruption, and defensin-1 immune peptide action occurring at the same time on the same wound surface. The multi-mechanism attack is the reason that, after 5,000 years of continuous honey application to bacterial infections, not a single honey-resistant bacterial strain has ever been identified in clinical literature. That is not a coincidence. That is the difference between a substance refined by 40 million years of evolutionary pressure to defeat bacterial pathogens and a single mechanism synthetic compound that a bacterium can learn to ignore within a generation of exposure. By 400 BCE, Hippocrates described specific honey application protocols for different wound types with a precision that modern wound care nurses would recognize as clinically sound. The Greek military adopted these protocols comprehensively. Alexander the Great's army carried honey as a standard medical supply across campaigns spanning 3 continents over more than a decade, treating battlefield wounds in environments where infection would otherwise have been universally fatal. Rome absorbed Greek medical knowledge and expanded its military application. Roman legion field surgeons carried honey in campaign medical supplies from Britain to Mesopotamia. A Roman soldier wounded on Hadrian's Wall and a Roman soldier wounded in the Syrian desert received wound care from the same substance, because the clinical evidence for its effectiveness was consistent across every theater of military operation. This was not one civilization's folk medicine. This was the universal clinical conclusion of every military medical corps operating in every climate across the ancient world for over 2,000 years. Wounds treated with honey survived. Wounds not treated with honey frequently did not. The knowledge moved intact through Islamic Golden Age medicine. Avicenna's Canon of Medicine described honey wound treatment protocols with greater specificity than any previous medical writer, documenting optimal honey types for different wound conditions and application frequencies. Medieval military orders, including the Knights Hospitaller, used honey as a standard wound care compound in field hospitals, treating Crusader army casualties across the Middle East. Every medical tradition on every continent that had access to honey arrived at the same conclusion. That is 5,000 years of convergent clinical observation confirmed independently across dozens of separate medical traditions, without any communication between them. The displacement of honey from mainstream wound care began in the 1940s with the commercial development of penicillin. The clinical efficacy of early antibiotics was genuinely extraordinary, and the medical establishment's enthusiasm was completely understandable. But the pharmaceutical industry that grew around antibiotic production was not neutral about a competing wound care modality that cost $6, required no prescription, produced no resistance, and in controlled clinical trials showed equivalent or superior outcomes for specific wound types. Raw honey cannot be patented. The mechanisms by which it kills bacteria are not proprietary. That equivalence is commercially catastrophic for an industry built on proprietary compounds. The FDA's 2015 restriction on health claims for honey wound care products completed the regulatory phase of the displacement. Medical-grade honey products with full regulatory registration do exist and are available by prescription through wound care specialists. They cost between $30 and $80 per application. The raw honey in your kitchen contains the identical active compounds at a fraction of the cost and cannot legally tell you that on the label. Now, here is exactly what raw honey does at the level of wound care chemistry that the jar in your kitchen is forbidden from describing. The antibacterial action of raw honey operates through 4 simultaneous mechanisms. The osmotic mechanism draws water out of bacterial cells through the concentration gradient created by honey's high sugar content, dehydrating bacteria faster than they can compensate through metabolic processes. The hydrogen peroxide mechanism releases a continuous low-level oxidative agent at the wound surface through the glucose oxidase enzyme reaction, killing bacteria through the same oxidative action that hospital sterilization protocols use at higher concentrations. The acidity mechanism maintains the wound surface at a pH between 3.2 and 4.5, which is hostile to the majority of pathogenic bacteria that require a near-neutral pH to replicate. The methylglyoxal mechanism disrupts the bacterial cell wall synthesis process through a biochemical pathway that no currently identified bacterial resistance mechanism can counteract. A 2011 study in the European Journal of Clinical Microbiology tested Manuka honey against 21 strains of bacteria, including multiple antibiotic-resistant clinical isolates. Every single strain was eliminated. No resistance to honey treatment was observed in any tested organism. A 2012 Cochrane database meta-analysis concluded that honey reduced healing time in mild to moderate wounds and burns compared to conventional dressings with significantly fewer adverse effects. Cardiff University researchers further documented that honey's multi-mechanism action penetrates and disrupts wound biofilm, the primary reason chronic wounds fail to heal through osmotic and chemical action that antibiotics cannot replicate. Here is exactly what to do, and the applications will reframe how you think about the jar that has been sitting in your kitchen as nothing more than a condiment. For wound care, use raw unprocessed honey or Manuka honey with a UMF rating of 10 or above for highest antibacterial activity. Apply a layer approximately 3 to 5 millimeters thick directly to the wound surface. Cover with a clean, non-adherent dressing to keep the honey in contact with the wound bed and prevent it from being diluted by wound fluid. Change the dressing every 24 to 48 hours for actively infected wounds, every 48 to 72 hours for clean healing wounds. The honey should maintain visible contact with the wound surface throughout the treatment period. For minor cuts, abrasions, and burns, a direct application of raw honey covered with a standard adhesive bandage is sufficient. The antibacterial action begins immediately on contact and continues throughout the dressing period. For infected wounds that have not responded to topical antibiotic ointments, raw honey is not a replacement for systemic antibiotic therapy in serious infections requiring medical attention. It is a direct replacement for topical antibiotic ointments in the wound care protocol for minor to moderate surface infections. The clinical evidence for its topical antibacterial efficacy against the organisms responsible for the majority of minor wound infections is stronger than the evidence for most over-the-counter topical antibiotic products. For burns, raw honey applied immediately to a minor thermal burn reduces pain, prevents bacterial colonization of the damaged tissue surface, and maintains the moist wound environment that burn healing requires. A 2015 randomized controlled trial published in the Burns Journal found that honey-treated minor burns healed faster and with lower infection rates than burns treated with silver sulfadazine, which is the standard pharmaceutical burn treatment used in emergency departments. For food preservation in field and off-grid conditions, raw honey's antibacterial properties extend to food preservation applications. Fruits, nuts, and other perishable foods submerged in raw honey are protected from bacterial and fungal spoilage by the same osmotic and chemical mechanisms that protect wound surfaces. Archaeological recoveries of honey from ancient Egyptian tombs have found the substance still edible after 3,000 years of storage. No synthetic preservative comes close to that stability at any price point. For sore throat and upper respiratory infection symptom relief, a tablespoon of raw honey taken directly or dissolved in warm water coats the mucous membranes of the throat with the same antibacterial environment it creates on wound surfaces. A 2012 study published in Pediatrics found that honey was more effective than dextromethorphan, the active ingredient in most commercial cough suppressants, at reducing nighttime cough frequency and severity in children. Dextromethorphan costs the consumer between $8 and $15 per bottle. Raw honey costs $6 for a jar that lasts weeks. One $6 jar of raw honey provides wound care capability that outperforms the majority of over-the-counter topical antibiotic products, burn treatment that clinical trials show superior to standard pharmaceutical burn dressings, food preservation capacity that requires no electricity or refrigeration, and respiratory symptom relief that peer-reviewed research shows more effective than commercial cough medicine. The Egyptian physician who packed honey into an infected wound in 2700 BCE and watched it heal when nothing else worked was not practicing primitive medicine. He was applying the most sophisticated multi-mechanism antibacterial compound available to him, which happened to also be the most sophisticated multi-mechanism antibacterial compound available to a wound care specialist in a modern clinical setting. The chemistry has not changed in 5,000 years because it does not need to. Bacteria have had 5,000 years to develop resistance to it and have failed completely. The pharmaceutical industry spent 80 years building a regulatory and institutional architecture that made you think of this compound as a condiment. The FDA restricted the label from telling you what military field medics use it for. The wound care industry priced its registered honey products at 50 times the cost of the identical compound in your kitchen to create the perception that medical efficacy requires medical pricing. A $6 jar has been sitting in your kitchen the entire time, waiting for someone to explain what the label cannot say. If this vault opens something for you today, subscribe and share this video immediately. Every share puts this knowledge in front of someone who will pay $40 this week for a synthetic wound dressing that performs worse in clinical trials than what is already on their kitchen shelf. Every subscription keeps this channel digging in the vaults they spent decades building walls around. The next vault opens soon, and what is buried inside is the one soil treatment method that turned barren land into the most productive agricultural territory in human history, that modern agricultural science has fully validated, and that the synthetic fertilizer industry has spent a century making sure you never connect to the dead soil in your own backyard.